It's my great pleasure to introduce Gail McConnell, who is our speaker for today. Gail is Professor of Physics and Director of the Center for Biophotonics at the University of Strathclyde. And she also leads the Mesolab, which we'll be hearing, I think, more about today. Gail studied optoelectrics and laser physics at the University of Strathclyde, where she also earned her PhD in laser technology. And her research now involves the development of new optical instruments for both cell as well as tissue imaging, including linear and nonlinear optics, the development of new light sources and sample preparation methods. Thank you, Gail, for joining us. We're really looking forward to your presentation. Please take it.
Okay, so thank you very much to the organisers for giving me the opportunity to speak to you today. Johanna's right, I'm going to be telling you about this Mesolab and the Mesolens, which is this giant objective lens for three-dimensional imaging with subcellular resolution throughout. So I'll start off by giving some brief introduction and motivation to the project. I'll then explain what the Mesolens is and how we use it as both a confocal or wide field instrument. And I'll conclude with some new and emerging applications and some detail on what we hope to do next, funding permitting, of course. So to kick off with, I realise I'm preaching to a whole load of imaging experts, and we're familiar with an image of this type. And this is not one of my data sets. This is from Kurt Thorne when he was at UCSF. And it's a fluorescence micrograph of one of these proprietary slides from Invitrogen or Life Technologies. And it's triple labelled with the DAPI omitted imaging of a mouse kidney section, where the green is effectively Alexafleur wheat germ agglutinin 488 and the red is Alexafleur 568 phalloidin. And we used images of this type all the time in our work, and it's great, it's absolutely useful. The big problem is when you're looking at a giant object such as a mouse kidney, because a mouse kidney is several centimetres in diameter. So that means that you can't normally study the whole organ with a light microscope, and you need to find some tricks to manage this problem. And the most common trick is stitching and tiling, and that's exactly what Kurt has done here. So he has taken an image of a region of interest using a microscope objective lens with modest magnification and numerical aperture. He's then moved the sample, imaged another region, and so on, and built up these little data sections or cubes, and then performed stitching and tiling using image registration software to make this larger kind of mosaic pattern of the tissue. Now, this is great because it means that we can see more of the organ than the light microscope can normally accommodate. But what it does mean is we have artifacts. So some of these artifacts are evident. So as an example, we have these dark bands that surround each one of these image panes. And this is a consequence of inhomogeneous illumination. You can, to some degree, counter that using flat field correction and so on. But there are other artifacts and problems that you don't see at this level of display zoom. And they normally manifest in registration of adjacent image tiles or image volumes. And it's a lot worse in 3 dimensions than it is in 2. And that can make image analysis, so for example, segmentation, if you're looking at a nucleus and it's disjointed over two adjacent panes, almost impossible. So while there have been great steps forward in stitching and tiling algorithms, we're not quite there yet. So we do still have problems with that method. There are other techniques that you can use to image large volumetric samples, such as micro-CT and optical projection tomography. And these are fabulous methods that should absolutely be considered when looking at tissue volumes that exceed a few cubic millimeters. Here's an example of some micro-CT images from the Long Lab. It was published in 2014. And I use this as an example because you can see how they've obtained images at different angles using a tomographic approach, taking the CT images through. This is a fairly late stage mouse embryo. They do that around the rotational point and then computationally make this 3D rendering of the tissue specimen. And then they can colour code organs or regions of organs as shown in panel C. The difficulty with some of these techniques is that they lack the high spatial resolution that's needed to often see some of the interesting cellular biochemistry. So you can potentially see a cell, you might be able to get subcellular resolution, but it's a bit of a challenge. So there's certainly further opportunity for development there. So we approach this problem as optical physicists do, trying to think about how we can use engineering and light to solve it. And we started off with what we knew about light microscopes. Now, what you see in this slide are the Rayleigh resolution profiles of objective lenses that you'll probably find in your own labs or own facilities. But these are in image space rather than specimen space. What you'll see is that irrespective of whether you have a high mag, high NA lens, or a low mag, low NA lens, or something in between, the Rayleigh resolution profiles look almost identical. And the reason for that is an historical accident. And it's because these objective lenses, and almost all objective lenses until very recently, have been designed with the visual acuity of the human eye in mind, because until quite recently, we've used the human eye as the photodetector, because you looked down the eyepiece and you sketched what you saw. Except we're not really in that space anymore. But that's not to say that these objective lenses don't have use. Of course they do. So, I mean, these flagship high mag, high NA lenses for immunofluorescence and immunohistochemistry work offer exquisite high spatial resolution, diffraction limited with sub-micron resolution in 3 dimensions, albeit within a tiny tissue volume. Then you've got the less expensive, but no less useful, low mag, low NA lenses, which can sample over large tissue volumes, but you don't quite have that subcellular resolution in 3 dimensions. So what we've done is overlook the fact that the human eye is involved in light microscopy any longer at all. So we normally use it only to crudely find the object, and then we move to digital photodetectors like photomultipliers or cameras. And so this has allowed us to think about a lens that has a unique combination of low mag but high numerical aperture. And what that gives us is subcellular resolution in 3 dimensions within over 100 cubic millimeters of tissue volume. So we can image hundreds of thousands of cells within a large tissue specimen. So we've called this lens the Mesolens because it sits somewhere between a macrophotography setup and a microscope objective lens. So it really is the Mesopotamia of the microscope world. So I can tell you a little bit about the Mesolens itself. So this diagram here on the left-hand side shows a cartoon schematic of an ordinary light microscope, the type you have in your own lab, where you put the specimen here, you have the objective here, you've got the tube lens, eyepiece, and a rather ineffective human eye as a detector at the top. This is a cartoon diagram of the Mesolens shown to scale next to it, and you can see that it's enormous. It's roughly the same length as a human arm, and it's roughly about as wide as a human arm, depending on how much you work out. So the specimen sits here, and you can see that we've got these large diameter objective elements that exceed 70 millimeters in diameter at certain points. And then we converge to an image plane that exceeds beyond the top of the screen here. And it's this that we've designed using Zemax. We then try and understand what we need in terms of glass, and knowing from experience that many of the glass preforms have dopant ions that are used for long-term stability and long-term use of glass types. We need to batch test the individual glass preforms before we buy them. So the image that's shown in the bottom here is taken from our trip to the glass factory in Wales, where we've put a UV laser pointer through 2 different supposedly BK7 tubes. You can see that one is strongly fluorescent in the kind of blue-green, one is weakly fluorescent in the orange. We can use neither of these because any fluorescence that results from excitation of the lens elements then makes its way to the image and would compromise our image contrast. So we have to discount those. We then, once we've found our fluorescence-free glass, basically reserve it, and we go back to the Zemax prescription, rerun it with now the cell-Mayer coefficients and Abbe numbers and dispersion properties and anything else that we need to know to ensure that our original Zemax prescription still holds true and will function. We then buy the glass, And because we don't have the capability to shape these bulk preforms into lenses at Strathclyde, we take use of a facility that's based outside London that also made the same, that made the end mirrors for the LIGO gravitational wave detection experiments. So we know that we're in good hands, and they shape the lenses to our needs, and then mount them in a central tube with a precision that's better than 3 micron in 3 dimensions. So they also have the mounting precision that we don't have at Strathclyde. They then give us the objective lens, and we turn it into a microscope by building around it. So what the Mesolens gives us is this low mag, low high NA combination, which is rather unusual. And I won't get through these numbers in turn because that's painful for everyone, but it does give us a 6 millimeter diameter field with a working distance that's around 3 millimeters, and that depends on the immersion fluid that we use. We have 2 or 3 correction collars, depending on what variation of the Mesolens we are considering. So we can accommodate imaging in oil, water, or glycerol. We can form an image in air, but it just doesn't have the subcellular resolution performance that's as exquisite as these other immersion fluids will give us. Roughly 90% of the effort in designing the Mesolens goes into chromatic correction. As a kind of postgraduate exercise, it's probably reasonably straightforward to design a monochromatic version that has fewer lens elements and is less complicated. But what we realised is that we are planning an imaging centre where people can bring their specimens. That means we need to accommodate the largest number of specimens that we can. So we have designed the Mesolens and proven its performance to be chromatically corrected across the entire visible spectrum. All of this comes at the cost of rich data, and rich data means large file sizes. So when we consider a single 2-dimensional image taken across roughly a 6 millimeter diameter field, it's around half a gigabyte. So then we throw in another couple of colors, we get to 1.5 gigabytes. Then we think about the Z direction for Nyquist sampling. In the Z direction, we need 1,000 images, so that takes us to 1.5 terabytes, and that's with a fixed sample. So that's why I say that the time taken to build an image is also variable, because we have 2 ways of acquiring data. One is confocal and one is wide field, and I'll go on to explain a little more about this now. So here is a photo of what the second version of the Mesolens looks like. Now, I say it's the second version because we have a prototype instrument that's shown in our original 2016 paper, and you can have a look at that if you're dead keen. But this is the one I think that probably best represents the state of the art. So this is the front part of the objective here, which extends into this black box here. One of the things that we wanted, despite having derided the human eye as a detector, we needed an eyepiece because no biologist should tolerate us waving a piece of white paper in the light path to find where our focal plane is and find the specimen for imaging. So roughly the footprint of this cube, with the exception of this part here, which I'll explain in a moment, is around a cubic meter. So it's not beyond reason that a normal lab can accommodate this. The confocal optics, which I'll explain in a moment or two, sit in this box here. We've got some lasers kind of out of field. And this long tube here is our camera port. So we normally have a camera up here. Said I would explain about the scanning system, but perhaps I should explain that really it is just a big confocal microscope. So we have a laser. We expand the beam using a Keplerian telescope. We collimate the beam using a long focal length lens, and we then put the beam into a pair of scanning Galvo mirrors. Now, ordinarily in a confocal microscope, the scanning mirrors are around 7 millimeters in diameter. Ours are around the same size as a car wing mirror because we need to backfill and overfill the back aperture of the scan lens, and that has an opening of around 60 millimeters or so, hence the car wing mirror-like approach. We use lightweight back-thinned beryllium mirrors, and they have almost enough good precision and repeatability. We have got problems with mirror jitter. We would like to solve them better, and if anybody knows of any other mirrors that we could use, that would be magnificent, or any other scanning approach, because that's one of the limitations that we have at present. But normally we bring the light through the scan lens, Mesolens, excite fluorescence, or look at reflection. Signal comes back up through the scanning system, back down through our 80-30 beam splitter, and we use that additional 30%, just 10%, just to give us more collection, so as a polarization effect. And we have a confocal iris and some type of filtration to block the laser, and a photomultiplier. We've got 3 channel detection and 1 in transmission. So here is some of the first data that we generated from our comparison with the Mesolens and a normal light microscope. So we used a Leica SP5 at the time, and we used a 5x 0.15 NA lens because it offered us roughly the same volume of capture and field of view as the Mesolens. The specimen here is a day 9 mouse embryo that's stained with the nuclear marker, effectively acridine orange, and it has been cleared in glycerol. So we image in XZ, so we're looking right through the centre mouse embryo here, and unsurprisingly, each one of these nuclei looks like a long thin needle of light. So that's a consequence of the low numerical aperture and the inverse proportion squared to the dependency on its resolution. So you can see the kind of macroscopic scale. So this scale bar is wrong. It should be 300 microns. Thankfully, we don't have a 300mm mouse embryo, or we should be deeply worried. But you can see, yes, we've got a large void here. We know that it's the cavity of the hindbrain, but it's very difficult to see almost anything else. When we put the same specimen on the Mesolens, prepare it in exactly the same way, this is the exact same specimen, mount it maybe slightly differently. And if anybody's particularly interested in how we mount specimens, I'm happy to chat about that later. Everything sharpened right up, so we can see now individual nuclei. We resolve features such as an indication, the neural tube with the blood vessels that now surround it, which are completely obscured in the ordinary confocal microscope data set. So we found this quite encouraging. That said, I realised that an XZ cross-section is quite a difficult thing to get your head around. So this is an XY image of a later stage. This is day 12.5 mouse embryo. The yellow contrast that you see here is the same acridine orange nuclear marker as we saw previously. The blue is an antibody label. It's an Alexa 594 conjugate to a neuronal variant of beta tubulin. And the full field of view here is 6mm in X and Y. So you can see that at this point in the depth in the specimen, we still have the macroscopic anatomy visible. We can see the ribs here. We've got some liver just appearing in shot, and the eye at this end here. If I press play in this movie, we then bring the specimen, so the Mesolens is upright, we bring the specimen closer to the Mesolens, and we're moving it in steps of about 3.3 micron over 60 planes. So we cover a total range of 200 micron in Z. And what you should hopefully see is that new information is being resolved with each successive plane. And that confirmed to us that it really is working as a confocal instrument. If I press play again, you'll see that these ribs disappear. You can see the brain coming into shot. We can see the heart starting to appear. And this shows us the macroscopic anatomy of the mouse embryo. Now, this is great, but it also hides the resolution that the data set gives us. So I've cropped out a region of the eye here in the next slide, and just computationally done some cropping and zooming. Now is probably a good time to explain that the Mesolens is not a zoom lens. So I realised that there are a bunch of manufacturers out there that will give you a stereo zoom macroscope, and they will purport to give you a combination of low numerical aperture and high numerical aperture and low magnification. And they do, but they just don't do it at the same time, whereas the Mesolens does, and we have a fixed relationship. We only have 4 times magnification, and we only have 0.47 NA. And any zooming that I'm showing you is entirely computational. So if we advance to the next slide, we can see this region of interest in the eye, and each one of these yellow dots is a cell nucleus. And if I press play in the movie here, you can see that the edge of the lens is shown here. This is the retina, and as we image deeper into the mouse embryo, it will collapse and go on to form the optic nerve. So we've used the Mesolens in confocal mode for a bunch of things, and I'll explain some of them later, but I did want to draw your attention to work that we published a couple of years ago now, where we developed a protocol for fixing, staining and clearing of whole, adult and third instar Drosophila, because they're one of the most important model organisms in biomedical research. So we published this image. It's been used worldwide because it's colourful and it looks recognisable to a lay audience, and that's great to see. The colours here are indicative of different depths within the intact fly. But I feel as if this actually hides a lot. So we're going to look at some raw data in the next slide. So this is a data set ported into ImageJ of Fiji. And we can use Fiji and ImageJ for a lot of our work, especially for small organisms like whole flies. If I press play here, we're just looking at me basically using Fiji and ImageJ to look at the data set. So we're going to pan and zoom around. So we're now in the abdomen of the fly, and you can see this is a female fly because these are the large polytene chromosome nuclei in ovaries here. We can simulate focusing and then come back up through the thorax. These are the fine rows of flight muscle adjacent to the legs here. There's a cardia approximately here. And then we can come up to the brain of the fly. You can see the edge of the lens of the eye here. And again, simulate focusing down. Eventually we reach some mouth parts, I believe, but you can see leg sections. We now have a holistic view of the fly, and we're working with researchers in the UK and Germany who are now applying the Mesolens to understand parasitic infection models better in their own research. So I'm going to switch quickly to what we're doing in wide field. And wide field presented us with a challenge. So the first challenge was we didn't think anyone would be particularly interested in using the wide field mode for Mesolens, so we kind of set it aside for a long time. But as we started promoting the work that we were doing, we recognised that actually there was merit in the benefit of the high numerical aperture, either for low light level imaging or just looking at fairly wide spread, wide fields of view, such as biopsy preps, but with the high numerical aperture that the Mesolens gives us. The problem was that we didn't have a camera. So because we, as I mentioned in one of the previous slides, we have around a 400 megapixel image, but we don't have a 400 megapixel camera. So at every trade show, we would ask the usual camera vendors, what do you have? And they'd say nothing. And then out the blue one day, we got a call from Teledyne Photometrics that said, we don't sell this, but another company does, and you might be interested, but you might not be. And it was really kind of them to just think of us. So we got in touch with Viworks. And this is a camera that's normally used by the automotive industry for assessment of defects in like car bonnets and doors. And it's a 28 megapixel sensor that's located on 2 orthogonal piezoelectric transducers. And rather than moving the sensor position by a whole sensor width, we move the sensor position by 1/3 of a pixel in X. We do this 3 times, and then we do the same, we move it in Y, and we build up an array of 3 by 3 images where they're just slightly spatially offsetted by 1/3 of a pixel in X and Y. Then we perform on these 9 images in total an operation that looks a lot like deconvolution, and we effectively then multiply the pixel number by 9, which it gets us to about 250 megapixels, and we end up with a pixel size that's reduced by a factor of 9. So while it's not quite the 400 megapixel that we would like, and the vendors now sell the 400 pixel camera that we need, we've just not quite got the funding yet to buy the thing in. If we reduce the field of view to about 4.5 millimeters, we're now Nyquist sampled, so we're in a good space. And we've used this for 2-dimensional imaging. So here's an endothelial cell prep, the blue or dapistine cell nuclei, the red or mitotracha red mitochondria, and the green is a fluorescein phalloidin stain. Now we know from some basic image segmentation in Fiji that there are 1201 cells within this field of view, and we can compare that to what you normally see with a light microscope, which is probably a few cells. But I quite like this data set because it shows one of the advantages of the Mesolens. So over here, everything looks great. So you've got lovely triple labeled cells and everything is good. Over here, things are less good. So you can see that something has happened. Now, either these are unhealthy cells and the F-actin has somehow polymerized or disappeared, or the fluorescein phalloidin dye just hasn't made its way to that region of the prep. So if you were a researcher using your local facility, you'd probably choose a region over here and publish that, but it might actually not be representative of the specimen itself. Whereas with the Mesolens, because we've now got this big field of view in high resolution, we're giving the researcher perhaps some bad news that they didn't want, but it allows us to reduce the subconscious bias that we all bring to our imaging experiments. But we can zoom in and see individual cell nuclei. We've got bundles of F-actin clearly visible, and you can also get a sense that there's some mitochondria in there too. We've also very recently used the wide field mode for imaging of thick and thin blood films that are part of a collaboration with colleagues in the UK and overseas. And we're specifically using mesoscopic imaging techniques in malarial diagnosis. So what we have done is a small comparison of bright field transmission with a normal microscope, Fourier tachographic microscopy. Then we've taken a convolutional neural network and performed some enhancement of our original low bright field images, and we've compared the lot with the Mesolens. And actually, it compares not too badly. But one of the most interesting outputs from this, which we're yet to build on, but is quite exciting for us, is that the convolutional neural network has been able to can recover or improve in the resolution of the raw bright field data, which has a numerical aperture that's almost identical to the Mesolens, to the point where we can see the individual Giemsa stained malarial parasites within the erythrocytes. So this gives us great hope that we should be able to do the same deep learning process and recovery with our mesoscale data sets, and it's probably just a matter of computational power to manage that. How am I doing for time? Okay, right, so as much time as I have left, I'm going to spend telling you what we're up to now and what we plan to do next. So one of the most recent things that we've done is imaging of large tissue volumes, much larger than the Drosophila and the mouse embryos that I've spoken about previously. And the first one of these is imaging of whole lobes of mouse lung that have been, this is a mouse model for tuberculosis. So we've applied a dual staining method that's fairly routine of auramine B and auramine orange and rhodamine B to stain up the bacteria within the intact tissue volume. So if I press play on a movie here while I'm chatting away, we can now image with the resolution that's required to detect through segmentation the individual bacilli within that intact lobe of mouse lung. So the contrast that you see here, hopefully these little orange dots, these are the bacteria. And we've developed, in collaboration with colleagues at the National Institute for Biological Standards and Control, methods we've now published about a year ago now for cluster analysis to understand the homogeneity or otherwise heterogeneity spread of the infection within this unusually large tissue volume. And at this point, because it's a point I'll probably come back to, I'll mention that this particular data set, which extends over, it's almost entirely 6mm by 3mm, 6 by 6 by 3mm image cube, took us about 2 weeks to obtain. So it's slow for us to image because we're moving, or because we need to sample over so many pixels. Even with a really short pixel dwell time in confocal mode, it can take us days or weeks to image samples. So it is slow. That's not to say it's bad, it just takes us a long time. But one of the other large tissue volumes that we've been looking at is ultra thick samples, specimens of mouse brain. So the cyan contrast that you see at this level of display zoom are DAPI stained cell nuclei. And if we, using Imaris, there's some rendering artifacts, but if we get past those and we zoom in to the data set, you can see these red blobs. So these red blobs are TD tomato expressing trypanosomes. So we're working with researchers at the University of Glasgow because we want to understand exactly where the parasites are. And that's very difficult to do without dissection because we want to know how they cross the blood-brain barrier, because that's still an open research question in this field. But one of the other features that we have here, using the exquisite resolution that the Mesolens is given is in 3 dimensions, is the ability to look at the parasite shape, because these parasites are, and I'll press play in the movie again, on the order of about 20 to 30 micron long, but you'll see, even depending on the orientation of projection, that some are long and some are short and stumpy. And that tells us about the stage of the life cycle of the parasite, and that in turn can tell us about the level and spread of the infection within the animal. So we're working with these researchers. We're now developing new tools that are open source for cluster analysis of these data sets, and we're looking to publish them in the next year or so. Working with researchers in my own lab, who are microbiologists, we've also been able to reveal new structures entirely. So the micrograph that you see on the left here is a mesoscopic image taken in wide field of a mature E. coli colony biofilm. So this biofilm is about 3 millimeters in diameter. And when you look at it with the naked eye, it looks smooth and dome-shaped. But when we look at it in cross-section with a Mesolens, because of the high lateral resolution, we see that we have an unusual kind of almost fractal-like pattern. And these are channels that run from the exterior of the biomass right into the heart of the colony. And we have been able to kind of map these out in 3 dimensions. So this is work that's ongoing. In fact, we've just submitted another preprint in the last month, because we know from the original work that these channels seem to, or do in fact, shuttle nutrients into the biomass. We know that these channels are used by the biofilm to try and sustain its nutrient supply and therefore its growth. And now what we want to do is understand, because we've used wild type, or at least a parental strain of E. coli here, which only has a GFP modification, we need to know whether the same happens in other E. coli mutant strains, which are found very often, especially in clinical environments. So that's the subject of our most recent preprint. And the next stage is to understand whether we can now use these newly discovered channels as expressways to shuttle antimicrobial agents into the biomass and therefore disperse it and get rid of it. So it may offer the possibility for new treatments and eradication. And when we're facing challenges in antimicrobial resistance, this becomes ever important. With the exception of the E. coli data that I've just shown you, everything so far has been fixed. And that's because of the data challenge that we have. But I'd like to show you something else that's living. So this is, again, unpublished data. This is a Drosophila Pupa. And the contrast in this image comes from GFP expressing hemocytes. So rather than using the sensor shifting camera that I mentioned in a previous slide, we've used the Hamamatsu Flash 4. So it reduces the field of view, but we gain an increase in the speed that's possible. We also suffer slightly from resolution decrease, but what it does mean is that we have, thanks to our very high numerical aperture, the possibility to image GFP expressing organisms at high speeds better than real time. So what we see here for the first time is a study of hemocyte flow in the intact living pupa. So you can see that we have this pulsatile behavior along the length of the pupa. We can see the pump at this end, and there's also a second pump at the top, which hopefully is not obscured on your screen. Now, normally the researchers we're working with in the University of Edinburgh are studying a region of view that's about this size, and that means that their sampling statistics are quite low. Whereas now we can image over very long, we're talking hours, if not, kind of approaching the day point, hemocyte flow, at least in 2 dimensions at the moment, and we have plans for 3 dimensions, so we can build up a better view of this important fluid flow. In my own lab, something, again, this is unpublished work, we're looking to get out in the next year or so. We're working with NHS Greater Glasgow and Clyde, and one of the problems that they have is tonsillectomies. And this is an extremely common operation. It's the most common paediatric surgery in the UK. And to my astonishment, we learned that the post-operative bleed in tonsillectomies is 20%. So you've got a 1 in 5 chance of being readmitted to hospital with a bleed if you have tonsillectomies. And this is unheard of. If most practitioners had recall rates of 20%, they would probably be under investigation, but it's seemingly normal for tonsillectomies. So we want to try and reduce the number of tonsillectomies that are required. And there's long been thinking within the community that bacterial infection may be responsible. And so if a patient presents with tonsillitis, they're given penicillin and told to come back if there's a problem. You'll only have your tonsils removed if you have 7 episodes of tonsillitis in 1 year, and that's outside of COVID. So you need to present often, and it is a debilitating operation condition. It affects children at important time points in their lives. It disrupts education and social well-being. So we want to try and reduce this. But there's almost nothing known about the effect of these bacterial infections in acute recurrent tonsillitis. So we've been working with clinicians in Glasgow. So we have a PhD student joined together who visits the surgical theatre as soon as the tissue has been excised. So she takes tonsils back to the University of Strathclyde, performs vancomycin blue dippy staining, and is then able to use the Mesolens to image bacterial biofilm infection in these excised tonsils. So I've pressed play in this movie. What you're looking at here is a reflection image of the tonsil surface in XY, and now we've just overlaid where the gram-positive bacteria are shown. So here, we zoom in, we can see that there's a bacterial flock present within one of the crypts of the tonsil because it has these ingress inlets similar to the invaginations in the intestine. And we can also see that there is a really high density of bacteria on the tonsil surface to the left-hand side. Where we're at now is building up statistics. So we need an N of at least 20 to consider what the patient outcomes are going to be. So this is just another sample to show a variation. Again, about halfway through the movie, you'll see that we overlay the bacterium, and this is widespread bacterial prevalence and burden across almost the entire tissue surface. We receive tonsils from patients that have either tonsillitis or sleep apnea. So that means that we have a disease and a control group. So we're looking to unblind in the next couple of months and then understand potentially for the first time really what the role is of a bacterial infection in acute recurrent tonsillitis. So I'm going to come to the last part of the talk now, which is kind of what we want to do next, or where exactly we're at right now. The first thing is that I mentioned it takes us days and weeks to image samples, and so often that's tolerable because if it's fixed, you press go, you walk away, you come back a week later, you save your data, and all is great. But for high throughput kind of longitudinal studies, it's not really feasible because your sample number needs to be high and it books equipment out for a long time. And we were asked for years, can we have a light sheet for the Mesolens? Every time we presented it and we explained the challenges, which are namely that we've got a 6mm field of view, we've got an axial resolution of about 4 micron, so we need a light sheet that's 6mm long and a light sheet that's about 4 microns thick. So Gaussian optics is not your friend at that point, because if you get a 6mm long light sheet, it's around 60 micron thick. So it kind of undoes the benefit of the Mesolens. But since we're supposed to know about optics, we set about trying to design and implement a non-Gaussian light sheet that would be compatible with the Mesolens. And we've now done this. So we have a light sheet that extends over the 4.5mm field of view of the camera that I mentioned previously, the sensor shifting device. The light sheet is about, I think it's about 7 microns thick. So it's still a bit thicker than we would like, but hopefully, I'll show you some data in a slide or so, it's convincing enough. The main advantage though is that we do seem to be able to preserve the spatial resolution so that it's comparable to that which we manage with confocal. And rather than it taking us, let's say, a week to build up a data set and acquire it, it takes us about, and I'll stop my alarm, it now takes us about 3 hours. So we're not at the high temporal speeds that the light sheet community are used to at the microscale, but for the mesoscale, it's probably getting up there. We're doing okay. I'm going to show you some, again, it's another screen grab of some raw data, because I prefer to show raw data wherever we possibly can. This is light sheet mesoscale imaging with the Mesolens of a whole mount of mouse pancreas. So we've got a whole mouse pancreas, you're only seeing part of it. And we've used a propidium iodide nuclear marker here. And what you're looking at is the data that we've just imported into the open source software Mapari, which is ideal for imaging our large scale data sets. And the student, Eliana Battistella, who's designed the light sheet and has used it with the Mesolens to study Katrina Wiesencraft's mouse pancreas preparation, has recorded the screen grab just looking around it in 3 dimensions. I'm going to push right to the end of the movie to show you the resolution that we have in kind of 3 dimensions. And we do have the ability to resolve individual cell nuclei within this large tissue volume. The last thing, the penultimate thing, is that we now have a lens prescription for a totally new Mesolens. So in response to what our collaborators and potential collaborators tell us, they like what we do, but they want to see more of the same. So that means an even larger volume of capture. So we now have a lens prescription, which we're having made in the hope of funding, please and thank you, that will cover the same area, so 6mm by 6mm in XY, but we'll be able to image up to a thickness of 11mm. And it will have the same resolution as the previous Mesolens through it. Rather than being chromatically corrected through the entire visible spectrum only, it will now be chromatically corrected up to the near IR, but the long wavelength end of the near IR. So we're looking at chromatic correction up to just short of 1100 nanometers. And the reason for that is that we want to perform multi-photon imaging. So that would be second harmonic, 2-photon, and possibly also 3-photon mesoscale imaging. One of the problems that we foresaw when we planned the original Mesolens work was dispersion compensation. But because we know the dispersion properties of each one of the individual lenses and its thicknesses, I've been able to calculate what the pulse dispersion is on propagation through the Mesolens at all wavelengths that we might use, and actually the dispersion is manageable, and so we're looking to go ahead in that manner. The lenses will be multi-immersion with water and oil as before, glycerol if we absolutely need it, and we'll have the point scanning and wide field that we mentioned, and now we have one, also light sheet. And then I'm going to finish up by showing probably the latest data that's come out of the lab. Again, obviously unpublished and raw. And this is mesoturf. So we've developed, using the Axelrod principle and some non-Gaussian illumination, a total internal reflection fluorescence mode for the Mesolens. So this now gives us a turf over a 4.4 by 3mm field of view. If I press play in a movie, so these are HeLa cells. We've got an antibody label for Paxillin, so we're labeling up the focal adhesions here. If I press play again, I've not done any segmentation in this, it's too new. I have no idea how many cells there are, but I think we can safely say there are a few hundred, and we can very easily see the focal adhesions, and we've got a much improved signal to noise ratio over what wide field can do for us. We've also been working with Rainer Heinzmann's lab, University in Friedrich Schiller in Jena, to try and develop a SIM mode, so structured illumination mode for the Mesolens. We've got some way along that path. We know what the limitations are, thanks to some work from a PhD student, Jan Schneider, and we are hoping to overcome those in the coming probably year or two. That's probably a long-term project. I also mentioned here your application, because we have this lens and it might be great for you, but you might not know, firstly, how to access it, or it doesn't have the mode that you need. We're always open to collaboration. And if the Mesolens project currently lacks some facility or mode that you would want to use, talk to us about it, because that's what informs our next stages of development. I mentioned that we have this MesoLab. So we have 3 Mesolenses in total that offer various combinations of wide field, light sheet now, and point scanning confocal, hopefully multiphoton in the future. We try and run this as low cost as we possibly can. We try not to charge for any proof of concept data. And so if you think that the Mesolens could be useful for your own work, get in touch with us, and we're more interested in what the science is than making money. So we'd be very happy to chat to you about how we could potentially support your mesoscale imaging work. So with that, I'd like to thank everyone that's been involved in the project thus far. So there are clearly a bunch of people at Strathclyde that have been involved both at the engineering, software end, but also in applications in biosciences. We have a number of UK and overseas collaborators. I've only acknowledged some of them here, whose data I've shown today, and also the funders, because without the cash to build the instruments and hire the people to do the work, we can't do anything. So I'd like to give them big thanks. And I'll conclude by thanking the organisers again for the opportunity to speak to you today, and you for coming along and listening to me.